All right, so in this section of Chapter 5, we're going to be talking about the different types of neurotransmitters that there are. So, to date, you've learned primarily about how a neurotransmitter would work. Well, there are actually different types of neurotransmitters that work in slightly different ways. So there are about 50 kinds of neurotransmitters that have been identified. Some are inhibitory at one location and excitatory at another. And while this makes learning about how neurotransmitters work more confusing, the overall message that you want to take away is that a neurotransmitter is not solely excitatory or solely inhibitory, that they are both, and that your body is really good at being efficient. And sometimes when you need to slow down at one activity, that means that you need to speed up at another. So in this way, it's really quite adaptive for neurotransmitters to be able to both be inhibitory at one location and excitatory at another. It's also important to know that more than one neurotransmitter may be active at a single synapse. And I'll be talking about this a little bit more later on in the chapter. The final element that's really important to know about neurotransmitters is there is not a simple one-to-one relationship between a single neurotransmitter and a single behavior. So there are a couple of neurotransmitters that are relatively common in terms of the fact that they've been heard of, one of them being serotonin, which is why I often speak about that in my examples, because lots of times it's been heard of before. So it's not that more serotonin is linked with a specific behavior. Serotonin has been linked with depression, for example. So it's not that more serotonin is better and less serotonin is more depression. It is not that simple at all. There's a very intimate and complex relationship with neurotransmitters and behavior. So there are a couple of criteria for what defines a neurotransmitter. So one, the neurotransmitter must be synthesized or present in the neuron. Okay, so it must be synthesized, made in the neuron, or be somehow present in the neuron. When released, the chemical must produce a response in the target cell. And this is really important. That basically, when you're sending a message, that message has some sort of response in the postsynaptic neuron. Another thing that's really important for having a molecule or an enzyme meet the criteria for being a neurotransmitter is that you can have the same receptor action when the chemical is experimentally placed on the target. Okay, this is really important. It wouldn't seem like why would this be so? But if you're to do an experiment to prove that the response that is exhibited by a postsynaptic neuron has to do with the neurotransmitter, then you should be able to produce that response in multiple ways. So you should be able to produce that response when the neurotransmitter is released from the cell, and you should also be able to produce that response when the neurotransmitter is placed experimentally on the receptors. And that basically, this is a way to prove that it is the neurotransmitter that is causing the response and not some other combination of factors. And finally, there must be a mechanism for removing the neurotransmitter from the synaptic cleft. And if there's not a mechanism for removing it, then the molecule wouldn't qualify as a neurotransmitter. So you have to meet all four of these criteria for a molecule or an enzyme to qualify as being a neurotransmitter. Neurotransmitters may also carry messages from one neuron to another by influencing the voltage on the postsynaptic membrane. Okay? They could also have a common message-carrying function, such as changing the structure of a synapse. And they can also communicate by sending messages from postsynaptic to presynaptic. So this means that the receiving neuron, the postsynaptic, could also be able to send a message to the sending neuron, the presynaptic. These reverse direction messages influence the release or reuptake of neurotransmitters. And these reversed direction messages can also be particularly important in drug effects, because a drug is going to cause an increase in neurotransmitters and sometimes or the release of these neurotransmitters. And so your body is actually going to try to compensate. So if you have a release of neurotransmitters in response to taking a drug, then you're going to have these reverse direction messages. These are delivered through autoreceptors, which I discussed in one of the previous videos. And that will say, hey, look, you're having tons of these neurotransmitters that are being delivered, way more than should normally be. So now what you need to do is, because you're having so much of it, you need to make less of it, for example. So if you make less of that neurotransmitter in the cell, remember the neurotransmitters have to be synthesized in the cell. So if you're taking a drug and it is causing an increase in the release of neurotransmitters, then your body is going to register that, and it's going to send back a message to say, well, look, there's too much of these. You should make less of them. And you make less of them, then you're actually going to need to take more of the drug to create that effect. So these reverse direction messages are really, really important for regulating the release of your hormones, and so that your body can self-correct itself. And they also play a very, very important role in habituating and adjusting to increase in drugs, so that as you take drugs, you're going to need to take more and more because your body will habituate to them over time. All right, there are 3 classes of neurotransmitters: small molecule neurotransmitters, peptide transmitters, and transmitter gases. And we'll be talking about each of them in turn. All right, so small molecule transmitters, this is a class of quick-acting neurotransmitters. These are typically the ones that pass from neuron to neuron in the fashion that we've been discussing so far. These are synthesized from dietary nutrients and packaged ready for use in axon terminals. So here's a little bit of a class question. So since, of course, you're not here with me, then I wouldn't expect you to answer this. But I want you to think about this for a moment. Why is the knowledge that small molecule transmitters are synthesized from dietary nutrients important to you? Why would you care about that knowledge? So think about it for just a second, and then I'm going to tell you why you should care. And if you want to, you can pause the video. If you don't, you can basically keep on listening. So the main reason that you should care about this knowledge is that the food that you eat is important for making the neurotransmitters that your body needs. And so these dietary nutrients, they don't just come from any food, they come from specific food. So part of the goal of a well-balanced diet, in which case you have greens and vegetables, possibly some protein, and certain types of fats, such as the good fats or the good cholesterol that you sometimes need versus the bad cholesterol, for example. So there's good fats and bad fats. There are specific nutrients and amino acids in fruits and vegetables, in whole grains. There's actually there's a little bit in meat, such as iron. So these are the types of things that your body needs to produce the neurotransmitters that it requires for proper function. And we don't actually eat for the taste of food. It's actually sort of evolved the other way, that the foods that we tend to, that are taste good to our body, it's more advantageous for those to taste good for us as well. So nuts and fruits and vegetables, this primarily applies to things in their naturalistic state. Sugars that would be advantageous, of course, fruits are primarily have sugars, and those have certain amino acids and vitamins that your body needs. And so the fact that those even taste better would cause us to try to seek those out. So the problem nowadays is that we have artificial sugars that hijack our taste to nutrient relationship system. So it is no longer that the things that taste good correspond to beneficial nutrients that our body needs, that we have bad fats that wouldn't be present in the naturalistic environment. We have artificial sugars, corn syrup, all things that would not be present if we were to eat the foods that we evolved to eat. And so these hijack our our nutrient in our digestive systems, we eat them, but they don't actually provide the nutrients that we need. We just end up storing them, or they convert to fat, or they can affect our digestive systems. So this is some of the real key reasons that you want to engage in a healthy diet. And this nutrient to food relationship is becoming increasingly investigated in mood disorders, behavior disorders, particularly in children who can be very, very picky with regards to food. So the goal is not to eat just till you get full. The goal is actually just to eat the items that contain the nutrients that your body needs for proper function. So here, for example, is acetylcholine. Acetylcholine is used very widely, so we need to synthesize acetylcholine. And to synthesize acetylcholine, we need choline, which is present when you break down good fats, such as avocado, olive oil, and salmon. This is the good fats that correspond to the good cholesterol, the good CDL that you would get for a physical. You also need acetate. Acetate is present when you break down acidic foods such as lemon juice and vinegar. So you need both choline and acetate to synthesize acetylcholine. These two important enzymes, acetylcoenzyme A and choline acetyltransferase, which would be most commonly known as CHAT, those two are important for or are required for acetylcholine synthesis. Then you also have the breakdown of acetylcholine, and when you break down acetylcholine, it gives you this enzyme, acetylcholinesterase, which can then be used again for acetylcholine synthesis. So for example, here you have the acetylcholine A carries acetate to the transmitter synthesis site. Okay, so you need this acetyl-CoA to carry the acetate to the transmitter site. You need both of them. Once it gets to the transmitter site, the CAT transfers acetate to the choline. This then forms the acetylcholine, the ACH. Okay, and this here is what your body needs. This is a neurotransmitter that your body needs on a regular basis. After this is released, the products can actually be broken down again into the acetylcholinesterase, the acetate, the choline, so forth. Whatever's not used in the synaptic cleft, they can be broken down and taken up and reused. Okay, so this is a really good example of why you need to eat certain items for proper protein or neurotransmitter synthesis. Another type of small molecule transmitter are these amino acid transmitters. Glutamate is a main excitatory transmitter, and GABA is a main inhibitory transmitter. So I said quite generally that on the whole there is no one neurotransmitter is all excitatory and all inhibitory, but they do tend to have one that is sort of overarchingly preferred over the other. In this case, glutamate is the main excitatory transmitter. It is not excitatory everywhere, but is excitatory most places in the body. GABA, on the other hand, is a main inhibitory transmitter. It is not solely inhibitory, but it is mostly inhibitory throughout the body. And these are just an example of what they look like on a molecular level. And finally, you have glycine. This is a common inhibitory transmitter in the brainstem and spinal cord. So these three are common amino acid transmitters that also make up that are also included in the small molecule transmitter group. Another important factor for small molecule transmitters is that they can be influenced by a rate limiting factor. And a rate limiting factor is any enzyme that is in limited supply, thus restricting the pace at which a chemical can be synthesized. So we just talked about some of the chemicals that are in acetylcholine. So if one of those were rate limiting, then it would be in limited supply. So only when that limited enzyme was there could the neurotransmitter be synthesized. So an example of a rate limiting factor relationship would be that you need tyrosine enzyme to produce L-DOPA, and then you need L-DOPA to produce dopamine, and you need dopamine to produce neuroepinephrine and epinephrine, which are used throughout the body, particularly in terms of movement. dopamine is used all sorts of places, but here you need this L-DOPA to make dopamine. And L-DOPA can be a rate-limiting factor. I'm going to give you guys an example of aspects of this process by looking at these two videos. This one here is an example of how some patients, their behavior is affected when they have a loss of dopaminergic neurons. And this one here features someone who has Parkinson's disease, and it shows a before and after of when before they have their L-DOPA and after they have their L-DOPA. And in this instance, L-DOPA is a rate-limiting factor. So if you can provide it with medication, then you can increase the rate at which the individual is able to make dopamine for use in their body. So now we're going to take a second to watch these videos.
Of encephalitis lethargica, or sleepy sickness. The disease, which killed millions at the time, causes lesions in the brain. Those who survived sank into a kind of semi-conscious purgatory that imprisoned them for decades.
For some, it was a kind of dream world, but others were condemned to tormenting repetitions, like the endless contemplation of 2 = 2 = 2.
One patient described how she had to walk around a mental square to 7 notes from a Verdi theme. And this would go on for hours and days, wouldn't stop. This, I think, is what one intuits, that there may indeed be something like a sort of hell.
Though he looks like a stray Santa Claus, Oliver Sacks is in fact a voyager in hell. He is a doctor who also writes about the dark continents of human illness. But he writes with such passion and joy, he has drawn millions of people into worlds they might have shunned. Like the world where the victims of sleepy sickness were still floating in emptiness back in the late 1960s. Sacks was a young neurologist at Beth Abraham Hospital in New York. He decided to gather the 80 known patients together, bring them in from the institutions where they had been stored like human furniture. He had heard about an experimental drug called L-DOPA, now used widely on Parkinson's patients. He wondered if L-DOPA might be the miracle which could jolt their brains back into life.
So I hesitated, and I hesitated for 2 years. They'd been put away for 40 years, and I didn't know what coming back to the world, and coming back to a world which was not their own, might mean to them. And then finally my hand was forced, because so many of the patients were terribly disabled. Some of them couldn't feed themselves. Some of them were dying.
It was 1969, and Sacks was right about L-DOPA. It was like seeing the dead arise. Up they came, up, out of the chairs that had been their prisons.
The first feeling was that it was just wonderful to be alive after being dead so long.
Esther, so frail she couldn't turn over in bed, walked for the first time in 40 years. And Rose, who went to sleep as a coquette, can be heard off camera singing a bawdy song.
If you don't tickle me in the right place, I'll take off my pet.
And then the flapper dances again.
When she came to in 1969, she kept talking about 1926 and talking about Gershwin and others who were current then. She said, I know it's 1969. I feel it's 26. I know I'm 64. I feel I'm 21. She said, I've been a spectator for the last 43 years.
Their skin was eerily young. Faces that have no expression acquire no wrinkles. And they were so greedy for experience, they bravely wrestled with the side effects of the drug. Yet for most, at the beginning, the jerks, the stops, were a small price to pay for life.
Lola Hester, who is having all sorts of side effects. and one arm is like this, all sorts of ticking movements, and she looks absolutely tormented by movements, and yet she suddenly smiles, and in that sort of gay, defiant smile, she's saying, you know, I'm alive, and damn you all, and I'm still enjoying life.
Lillian Ty wrestles with one of the side effects of L-DOPA, an uncontrollable shaking of her head.
What's it like doing your hair when your head's shaking like that?
Kind of follow it around. Devil putting a part in, though.
When the medicine wears off, she sits frozen like a stone. But 4 times a day, she gets L-Dopa, which she relishes like a cocktail in the desert. She had just taken it before we walked in the door.
With the first medication, what was the first new sensation you felt?
Move.
You could move. It was a miracle, yes?
Yeah.
In an inexplicable quirk of the brain, L-Dopa causes Lillian to speak in repetitions like a slightly scratched record. Yet when she reads, it stops.
She didn't go to the hospital to have an oxygen test.
And it stops when she sings.
It will get started now. I can't afford it. And I'll be there if I'll declare none of us will go for two.
And even when she's repeating herself or hurled around by the medication, she recruits you to laugh with her at her quirky, determinedly happy life.
I didn't realize there would be this sort of resurrection in the summer of '69, and I certainly didn't anticipate how grim things might become afterwards.
Within a matter of days, it was clear. In some patients, the drug not only triggered strange gestures, but grotesque appetites for sex, for food.
And sometimes after eating, to sort of stuff their fingers on napkins into their mouth, and they felt they were losing control then, and losing dignity.
Some of the patients decided it was better to have dignity in purgatory than life without any pride. Leonard asked to be taken off the medication. Within hours, he returned to his frozen state, bright eyes inside a concrete body. And in the end, Rose, the reawakened flapper, couldn't accept the 64-year-old body trying to dance to those girlhood memories.
She said she didn't like our television age, as she called it, and after 10 days of this sort of strange 1926-ish animation, she rather suddenly went back to this trance-like state, and nothing we could do after that sort of had any effect on her.
Did they seem failures to you?
They seemed realists in a way. I mean, there's great dignity, great stoicism in this position without hope or without regret. I think finally it's a message of survival, that one can go through hell and one can be in hell and yet survive and even transcendently as tough and funny and loving life, even if there is no hope in the ordinary sense.
Say I love you, if you seem to tell a lie.
Oliver Sacks and his patients have reminded the world of a devastating story. Starting around 1917, the sleepy sickness epidemic crisscrossed the world, leaving almost 5 million people dead in its wake. In 1927, it essentially disappeared, although many institutions had to be built just to house all the living victims who were left permanently damaged.
Okay, now here's a very different film about a person with Parkinson's, and you can see the differences in their behavior and movement before and after they take L-Dopa, which is a rate-limiting factor. And after this video completes, then I'll talk with you briefly about both of the videos.
In this first clip, we're examining the gait of a 55-year-old man with Parkinson's disease during the off phase. Notice his typical Parkinsonian gait with shuffling and elements of festination. In addition, notice his reduced arm swing.
Typically when people walk, they have a bit of they swing their arms.
Notice how difficult he finds to turn when asked to do so by the examiner.
You see, notice he doesn't swing his arms at all.
In the second clip, we're looking at the same patient about 30 minutes later after he's taken his L-Dopa medication. There is a dramatic improvement in his gait, but if you look closely, He also demonstrates some mild dyskinesias of his head and neck as his head wobbles a little bit from side to side.
So both of those videos give you a very dramatic illustration of what can happen when you don't have access to a neurotransmitter or an enzyme, when you don't have access to an enzyme that is a rate-limiting factor in the synthesization of a neurotransmitter. In this case, it's dopamine. I do want to speak briefly about Oliver Sacks as well. A couple of you some of you may have heard of him. So I would greatly recommend that you look into some of his books. He has a couple that are quite famous. Awakenings is a book that's written by Oliver Sacks that was later made into the movie with Robin Williams. And so I very much recommend both. He also has another famous book, The Man Who Mistook His Wife for a Hat. But they both, you get a really entertaining glimpse into elements of neuropsychological disorders, but you also have the potential to learn a great deal as well. So I highly recommend him if you're interested in this type of material and content. And to turn to the other patient who had the Parkinson's, you could see the very dramatic change in his behavior and his gait as he walked. Both of the sleepy sickness or the encephalitis lethargica as well as Parkinson's, the loss of movement would oftentimes be things that were associated with purposeful movement. So or goal-directed movement, they have fewer issues with automatic behavior. And so even if you noticed in one of the sleepy sickness patients who had decided to go off the medication, it showed him lying there motionless, but he would still be blinking. So blinking is a type of automatic motion. Of course, it still requires muscles, but it is so automatic that even if you have huge problems with muscle movement, those muscle movement problems tend to be focused on purposeful, goal-directed, voluntary movements rather than involuntary movements. And the brain is actually organized very differently for voluntary movements versus automatic movements. Automatic movements, eye blinking, and even automatic muscle movements such as your heart beating and breathing, those tend to be controlled by different regions of the brain. So you can have the maintenance of automatic movements, but the loss of voluntary movements. All right, so in addition to small molecule transmitters, you also have peptide transmitters. So peptide transmitters are known as neuropeptides or neuromodulators. Peptide transmitters are a multifunctional chain of amino acids that act as a neurotransmitter. They are synthesized from messenger RNA based on instructions from the cell's DNA. The synthesis of peptide transmitters is slow, and peptides also act slowly and are replaced slowly. Importantly, peptide channels do not bind to ion channels. They do not have direct effects on the voltage of the postsynaptic membrane. They tend to act indirectly. Okay? So peptide transmitters activate synaptic receptors to indirectly influence cell structure and function. Talk in the next video about the differences, different types of receptors. And so when we get to talking about different types of receptors, then I think you'll actually have a better understanding of how peptide transmitters are different, for example, from the small molecule transmitters. Peptide transmitters typically, because they are slow of synthesis and they eat slowly, because they're slow of synthesis and they act slowly, they typically occur in the body as hormones, and they regulate eating and drinking and pleasure and pain. Here's some examples. So opioids are peptide transmitters. Insulin, that you would have heard of a great deal. Somatostatins are a peptide, and you have corticosteroids. Corticosteroids are very important for regulating the stress response. Corticosteroids are secreted in times of stress. So all of the neurotransmitters, small molecule transmitters, or peptide transmitters, all of these, look at some of the examples that we provide in the tables in the book, because you'll see or you'll read the names of neurotransmitters that you should be familiar with or you would have heard of. And finally, we have transmitter gases. So an example of a transmitter gas is nitric oxide or carbon monoxide. Neither is stored in synaptic vesicles nor released from them. So carbon monoxide, your body uses that often, and one of the reasons that carbon dioxide is so poisonous is that carbon dioxide can take the place of carbon monoxide neurotransmitters, and in replacing them can severely inhibit the body's function, and it can be one of the reasons that you can die from carbon dioxide poisoning. Nitric oxide or carbon monoxide, they are not stored in synaptic vesicles nor released from them. Instead, they are synthesized in the cell as needed. And because they are transmitter gases, they can easily cross the cell membrane. Their effects include controlling muscle lining and intestinal walls and dilating blood vessels. So one of the things that I'd like to point out from these effects is that both of them are relatively automatic. So automatic behaviors that your body controls for, dilating blood vessels, muscle lining of intestinal walls. So to give you an example, common medication that targets a transmitter gas would be Viagra. So the nitric oxide, that dilates blood vessels in active areas of the brain and in sexual organs. So nitric oxide is active in penile erections. And what Viagra does is Viagra acts by enhancing the chemical pathways influenced by nitric oxide. Now you have a little bit of a better understanding of this common medication that you see all the time on TV. And it says if you were to have a reaction to the medication, for example, that lasts more than 4 hours, then you would want to contact your doctor. Well, you can see why this would be real problematic. If Viagra acts by enhancing the chemical pathways influenced by nitric oxide, and nitric oxide dilates blood vessels, so if you've had your blood vessels overly dilated for more than 4 hours, that's going to be dangerous for your body. And so now we're going to transition to a new video in which we begin to talk about the different types of receptors that receive neurotransmitters.